Great. Well, thanks so much, Jonathan, and really nice to see everyone. So I'm James. I'm a faculty at Biomedical Data Science. Been at Stanford for I guess around 9 years now. I guess maybe one kind of fun fact about myself people might not know is that I used to be to work for different newspapers. Used to be like work on different journalism things, and probably the most fun things I did was actually to review various theater and movies and restaurants for different newspapers. So maybe it's the next colloquium. We'll talk more about that. Okay.
Was that a hobby or was that a job?
It's a very good job that became a hobby. Yes.
You know what app he uses so I can follow his restaurant recommendations.
Okay. Sorry, once said what app to follow your restaurant reviews?
Oh, so I haven't reviewed these things recently. So that was for these newspapers many years ago. Yeah.
All right.
I know in the room where we have a room microphone, so we're going to mostly keep the room on mute so there's not too much background noise. I'll let Excel kind of take it over from here. If questions come up in the meantime, we'll interject. But otherwise, why don't you go ahead and take it away? We've got quite a call. We have over 96 people online and another 40 plus in person. We're all very excited to hear this discussion. Take it away.
Okay, great. Thanks, everyone. So I want to share some recent directions we've been exploring around building these different kind of AI scientists to help us to accelerate different types of biomedical discoveries. So the broader context of this is that we as a community has been building a lot of AI tools for biomedicine. And usually in the past, when we develop an AI tool, you start with a well-defined problem, like a specific problem, and then you develop a tool to tackle that problem, right? So for example, if your problem is to predict structures of proteins given sequences, then AlphaFold is a very powerful tool for doing that. In the last year and a half in particular, there's been kind of an interesting paradigm shift where now people are, instead of thinking AI as a tool for solving a well-defined and specific problem, they're thinking of AI as sort of like a co-scientist that can potentially help us across much more diverse kinds of research endeavors, right? All the way from coming up with hypotheses, research plans, to experimental designs, and so on. A lot of that shift is driven by what people call these AI agents, which are essentially systems that you build on top of large language models that then enables these language models to use different tools like AlphaFold, but also to communicate with each other and also communicate with other AI systems and human researchers. Right? So that enables these agents to potentially act almost like a colleague that can help us across this broader range of research endeavors. So I think we're still in quite early stages of that paradigm shift, but it is quite exciting, quite interesting. And what I want to do today is to share sort of 3 examples of things we've been working on with my students that to illustrate the kinds of things we can do with these new types of AI co-scientists. And I'll pause after each one of these, so feel free to ask questions throughout. So the first example I want to share is this platform that we built recently called the Virtual Lab. It's basically a platform where we have a team of different AI scientists work together to tackle research problems. So very much mirrors my physical lab at Stanford, right, where there's like an AI professor agent, and then working with the AI professor agent would be a bunch of different AI student agents with different expertise, right, in data science, computational biology, immunology, and they have their own group meetings. And we also have like a mechanism, an agent school, through which the agents can actually teach themselves how to become a deeper expert. So this work was recently published. It's led by Kyle Swanson, who I think is actually in the room. So if you have questions, he would be a great person to talk to afterward. So I'll first explain a little bit how the virtual lab interaction look like, right? So when we interact with these different AI agents, we mostly do that by talking to the AI PI agent. So for example, I can explain to the PI agent like some description of the problem I'm interested in. Maybe I want to design this kind of protein or analyze this complex genomic data set. And then the PI agent will look at each of these problems and say, okay, what are the different areas of expertise that are needed to tackle that project? Maybe for this particular project, it's useful to have a computational biology on the team, but then the PI agent would actually automatically create and then spawn that agent that have that relevant expertise. So I'll come back to in a couple of slides to explain more of how the training and of the agents can happen. But for now, you can think of each agent as basically being like a large language model under the hood. So here we can use GPT-4o. You can also use GPT-5 or other models. And then we can endow those agents with more specialized knowledge and also more specialized tools that are relevant for their domain. So after the agents are automatically created and trained by the PI agent, then they can start to run their own group meetings, right? So the agents will have their team meetings where they'll meet together to discuss and come up with a research plan, similar to our human team meetings. They can also have one-on-one meetings where one of the agent will actually meet with the PI or the critic agent and to review a sub-project, right? Maybe analyzing a piece of a code or reviewing a specific subtask. So here, just to make things a bit more concrete, here I just want to show an example of what this kind of agent team meeting or group meeting will look like. So this was actually the first project we assigned to the virtual lab, which is basically to help us to design new binders to recent COVID variants. So at the time of this project, so there were actually a bunch of these new COVID variants started to spread last year, and people didn't have existing good binders to the variants. So we thought this is kind of a timely, but also quite challenging open-ended research project that would be important if the agent could actually help us to tackle. So in the beginning of this group meeting, right, so the PI agent spoke first and says, okay, here's the overall objective, the goal of this project is to design binders to the latest COVID variant spike protein. And then the different sub-agents can then speak up sequentially to contribute their research ideas. Right, in this case, the immunologist agent spoke up next and says that, okay, I recommend actually design nanobodies. This is already maybe a little bit surprising because if you ask most human researchers, they might say, okay, let's design antibody binders, which are much more common, more widely used. So nanobodies are basically small cousins of antibodies. They're found in only a few animals like camels. So this may be kind of an unorthodox recommendation to go for nanobodies rather than the more common antibodies. But there turns out to be actually a good reason for this, as we'll see next, right? So the machine learning agent actually spoke up next after the immunologist and says that, okay, I actually like this idea of looking after nanobodies. Because these nanobodies are smaller proteins, that actually makes my job as the machine learning agent easier because for me to computationally model and predict their structures using deep learning methods, right? So you can see how different agents can sort of build off ideas of each other. And then in all of these meetings, we also have a critic agent whose job is to basically provide more skeptical feedback to those other agents. So here, the scientific critic agent says that, well, there's not a lot of public data around nanobodies for COVID, so you need to be quite careful about overfitting your predictions, right? Especially if you're building these deep learning models. But the actual meeting goes on for longer. Here, I'm just showing you sort of the beginning of this group meeting, but you can already see how the agents can interact with each other, and it can also build ideas of each other to come to some consensus of the research plan. Great. So there's several benefits of these agents, but one thing that makes me very envious of the AI agents is that their meetings are much more efficient than my meetings, right? So one of their meetings maybe will take a few seconds or less than a minute. So in the time that we had one of our meetings, they can already have hundreds of research discussions. And because the AI scientists, they don't get tired, they actually run multiple meetings and discussions in parallel. So basically for every scientific decision that they want to make, we can run 5 meetings in parallel. They will discuss the same topic in these parallel meetings. Because there's some randomness in these language models, right? So in these different parallel meetings, they can often come up with different ideas. And then we ask the PI agent then to read the summary, the transcripts of all these parallel meetings, and then come to a consensus of what to do next. Right? So it's kind of an effective way to explore a broader range of ideas and then come to a consensus and still have a concrete set of to-dos. And in case you're curious, here I'm showing this pie chart, basically how often the different participants would speak in the virtual lab discussions. Right? So for example, the human researchers, like myself, my students, like the users of this virtual lab, we don't talk very much, maybe about 1% of the time. Right? So our job is to mostly provide for some high-level guidance to the agents. Right? For example, we can tell the agents, here's the project that we're interested in. We can provide them some constraints, like here's how much money we have to do the experiments. We can also provide some feedback, like if they make mistakes, we can tell them, okay, you should try something else. But otherwise, we really want the agents to basically do their own work and implement and design their own projects, right? So we don't want to micromanage the AI scientists too much. Here, the computational biologist does the most talking, which I think makes sense because here the CompBio agent actually has a lot of work to do to sort of design the computational pipeline for optimizing the nanobodies. The machine learning agent does the coding for the other agents. So another agent might say, okay, implement this algorithm, and then the ML agent actually write that algorithm in code. So it's also quite important. So now that we've seen how the agents can interact, I want to come back to and explain various ways we have for actually improving and training the agents. Right. So the basic version of this virtual scientist is that you can just sort of take the existing models, frontier models like GPT-4 or GPT-5, and then give each of these models sort of specific instructions to be different kinds of AI scientists. Right. And that actually works reasonably well already. These frontier models, they have quite broad knowledge, but sometimes if you go really deep into a particular domain, then these models may lack like the latest insights from the latest clinical trials or latest viruses and so on. Right. So to fill in that gap, then we have this agent school where the agents actually go to the school, right, and then teach themselves how to become a deeper expert, sort of like a school, sort of like a replica, like a virtual Stanford. All right, so the way that the school works is that we'll tell the agents here are some of the topics that are important for you to learn about. Like one topic could be like learn about the latest COVID variants or learn about the latest AlphaFold. And then for each topic, the agents will come up with their own learning curriculum by generating a set of questions that are relevant for that topic. Based on each of these questions, then we enable the agent to be basically able to go out and search and download papers that are relevant for each of these topics. They'll find these papers maybe by doing like PubMed search and finding open access articles. They'll download these papers and then read them in depth. As they're reading the papers, then we also allow the agents to basically update their model parameters, right? So under the hood of each of these agents is a large language model, like a transformer neural network. So then as they're reading this information, then the agents can actually do supervised fine tuning to update the weights of the transformer to help it to ingest more information. And then we also have an assessment mechanism where like some teacher agents will generate quizzes to test how well the student agents have learned a particular topic. If they don't do very well on the quizzes, they have to go back to school to additional learnings, readings, and trainings before they can graduate from the agent school. Right. So the agent school is also a relatively fast and efficient process. Usually within hours to a couple of days, the agents should be able to actually learn quite a bit of these new topics. So we find that this is, in our assessments, this can be an effective way to help the agents to learn some of the more up-to-date knowledge. Okay, so now that we've seen how the agents can interact and also how they can improve, I want to come back to this running example to illustrate the kind of research that these AI agents are able to do. So recall that the first project that we give to the virtual lab is to basically help us to design binders to the recent COVID variants, and they decided to design these binders in the form of nanobodies. Now, because there's not a lot of public data for nanobodies, so the way that the agents try to overcome that data shortage is to basically come up with a computational workflow to help them to in silico optimize these nanobodies. So here's the workflow that they designed and implemented. It's kind of interesting. The first, the agents use this protein language model called ESM to basically help to filter for high quality candidates. Right? So ESM is basically takes its input, some amino acid sequence, and then computes a score to see how likely is that sequence to form a stable and functional protein. Right? So then the top candidate from that, then the agent would pass that into AlphaFold together with the spike protein of the target COVID variant. And AlphaFold generates this three-dimensional structure of how the candidate antibody might bind to the target. The agent will look at that three-dimensional structure and extract some scores to see how compatible is the binding. And then we'll pass that three-dimensional structure into Rosetta, which does more like a physics-style relaxation of that structure and then estimates the binding affinity. So these different tools used by the agents each provide some complementary information. The agents then came up with like a holistic scoring mechanism combining the insights from these different tools and use that to basically prioritize the variants, the nanobodies, and iterate through this pipeline multiple rounds to look for the most promising candidates. So each of these tools have been previously used, certainly in previous publications, but this workflow that agent created is, I think, somewhat different from what people have published before. There's some subtle differences, for example, in how the agents actually uses ESM compared to what people have done before. So the agents are able to have some of these combinatorial creativity in taking existing tools and combining them in sort of like a new way to adapt it to a sort of a new problem at hand. So this is all done by the agents, right? So this is all in silico. And since the AI scientists, they don't do their own physical wet lab experiments yet, right? So they basically give back to us, like here's a list of 92 candidates that the AI agents thought was the most promising. And then we, with our experimental collaborators at the Chan Zuckerberg BioHub, then we actually would synthesize these 92 nanobodies and then test them in the wet lab. So here are two example nanobodies designed by the agents. These are new nanobodies. They're quite different from any existing nanobodies. And the bottom basically shows the wet lab experiments to test how well do they bind to different COVID strains. So I'll just explain what each curve means. So if we look at the bottom red curve with the open red circles, that's where we take existing nanobodies that were previously designed by human researchers and test that against a recent COVID variant called JN1 variant, right? So it does not show very good binding, right? So it basically stays around 0. The second red curve with the solid red circles is a nanobody designed by the virtual lab agents. And now it does show more promising binding. And then these two blue curves are the nanobodies. If we test these two nanobodies against a very different strain of COVID, the one from Wuhan from 5 years ago, you can see the AI designed nanobody, the top one also shows more promising binding compared to the previously human expert designed nanobodies. There's definitely a lot more experiments that needs to be done to further characterize and validate these binding assays and these nanobodies. And we've actually also given these results back to the AI scientists themselves, so they have further improved and optimized their own computational pipelines. And I think this already kind of demonstrates some of the quite exciting potentials that within a few days, the AI agents are able to come up with kind of a sophisticated computational workflow, implement it, and then help us to really prioritize the designs and optimizations of these nanobodies, which then we can experimentally validate. I'll just wrap up this part here and then open up for some questions for the part 1. So here we actually released the virtual lab, so it's all open source as a part of our recent paper. And people can now use this to try to tackle quite different kinds of research projects. So we have also other colleagues at Stanford who are using this to look for biomarkers for Alzheimer's disease, for example. So it's not really limited looking at protein tasks. We did experiments where we showed that actually when you have teams of multiple agents with different background that work together in these groups, that often is better than having a single agent try to tackle the problem by itself. Basically, what happens is that when you have multiple agents, you often sometimes have disagreements and have to resolve those disagreements and debates. And that often leads to more creative and more robust, more interesting reasoning than if you try to have a single agent to do this task by itself. We also found it's quite useful to provide the agents with some sort of resources. Right? So for example, it's useful for them to have some of the memory, like a digital notebook, so they can remember what did they talk about before and then the previous group meetings and discussions. And also it's useful for them to have like a virtual environment, like a sandbox, so they can safely locally run these different tools like AlphaFold, and also safely modify these different tools like ESM. And finally, I think the agents also have their kind of interesting social dynamics, right? Similar to how human researchers, like, when we have group meetings, some people talk more than others, and that changes the dynamics and the ideas. In the agents meetings, depends on who speaks first, and second, that also changes the meeting dynamics. But something that's easier to do with the AI agents and hard to do with the humans is that since we can run multiple meetings in parallel, right, we could actually have different agents speak in different orders in the parallel meetings. You can also assign different personalities to different agents, and almost use this virtual lab as kind of a petri dish to study organizational optimization, right? What combinations of scientists and personalities leads to the most creative and most robust, effective science. I think there's also, or I'm quite excited about using this as sort of this kind of computational social science experiment as well. So I'll pause if people have any questions about this first part before I move on to other examples of agents.
So James, I see there are a handful of questions in the Q&A box. I wonder if you want to jump on some of those and see which ones you want to answer now or address later. Thanks.
Okay, yeah, so I just opened the Q&A box. Yeah, so there's a few questions. So Steve has a question around basically how are different randomness created introduced, right? And also, how does human participate in these meetings? So these are good questions, right? For the human participation, so basically there are different parts where the human researchers can give feedback, right? So often what we do is to basically have the agents like summarize their discussion. So we don't need to look over everything that they talk about. Maybe have the PI agent to summarize their discussions and the research plans, right? And then we can tell the PI agent, oh, like, I think this is a good idea, or we don't like this idea, or try something else, right? We can also have one-on-one meetings with the agents, right? So we can, for example, meet with the machine learning agent and then have them review the piece of the code that they wrote and then help them to debug the code, right? So it's similar to how like an advisor, a human advisor might advise like a human student, right? So that's similar to how we can advise the virtual lab agents. And then there's also a question around like who actually implemented the workflow. Yes, so the agents, they designed this workflow, right, using the ESM and AlphaFold and Rosetta, and then they actually do their own implementations, right? So this is where, for example, the machine learning agent actually write to the code, right? They do all the code. So we don't do the codings for these agents. Okay. Are there other questions from the room? Also, feel free to also just ask live. That's also fine.
Yeah, I actually had a question kind of building off the first one. So, and also this last bullet on, you mentioned how you could have multiple meetings to take account for the dynamics of who speaks first and who follows and how that changes the findings and direction of any particular meetings. I really like that point. The thing I was wondering though is, for the human researcher, you mentioned that sometimes they need to or will provide important course correction or parameters into those meetings to guide those agents to make sure that they don't get off the rails. So like, how does the human then interact with multiple meetings? Like, like especially if you need to add in that tidbit earlier on, and then they're interacting so quickly that if they're already on like the equivalent of human hour 1, and then you're like, oh wait, by the way, you forgot in the fourth Thursday of the meeting to account for our budget is only $10,000 for this phase.
Yeah, so I think sort of there's definitely a lot of room for thinking about how to really design the human AI interaction here. Right? As you mentioned, they work at different speed and different schedules. Right? So I think there's a lot of creativity here. So we did something relatively basic. That's we can look over that are summarized by the PI manager agent, right? And then we can periodically basically provide feedback to the PI agent. You can also imagine other ways of working with the virtual lab where you could try to look over their shoulder more frequently and then provide more regular updates. And if they have multiple meetings, I think it's often quite maybe too much for us to follow all of the meetings in parallel. But we can basically still have like the PI agent to summarize the main output from each of these parallel meetings. And then so we can then take a quick look at that and say, do we want to provide any feedback?
So I have two questions. So first, how do you adapt the agent? So how do you make an agent a computational biologist and immunologist, and how do you give them personalities? The second question is, do you only use purely generative models or also like reasoning models?
Yeah. So these are good questions. So here the virtual lab is compatible with different kinds of models, right? So we could actually use reasoning models or the latest models, right? And I think in some of the more recent applications for different collaborators, they're also using different versions. So you can use reasoning models. In terms of each of the agents, like I think a big part of the agent is that they have for their own particular domain, they have their own literature and also their own tools. Right. So for example, for a computational biologist, it's actually very useful to be able to use tools like AlphaFold, right? So that's where we provide those tools in like a local environment for that agent to use. If it's like an immunologist agent, then it's very useful for that agent to learn about like the latest developments in COVID, right? Like the latest COVID variants. And that's where we enable the agents to basically read and download the relevant literature and be able to update its own knowledge about the latest information.
Okay, no more questions, James. You can continue. There's a few on the.
Okay. Okay, great. So I'll go on a little bit more and then I'll pause again and then we can have more broader discussions at the end as well. Thanks for the great questions. Okay, so following up on that on the virtual lab, so recently we've also been really taking a deeper dive into, I think, some specific areas where we think these AI agents can be quite useful. And I think actually data science is one particularly really promising area where these agents can really help us. So we've been building this data scientist agent that we call Cell Voyager, which can take as input quite complex data sets, which could, for example, be like transcriptomics or genomic data sets. And we actually generate these Jupyter notebooks, right? Multiple notebooks. Each notebook responds to like an in-depth analysis of the data. And then it will summarize the findings from these multiple analysis, right, into a set of figures and interpretations to help us to analyze and make sense of these complex data sets. And the reason why I think is, data science is a particularly good application area for agents is that, as people here know very well, is that in modern data science, often we're dealing with increasingly larger, more high throughput, more complex data, right? So that's already quite a big challenge. And in parallel, right, so now there's also this really rapidly growing ecosystem of different tools, right? So now there, for example, just in transcriptomics analysis, there are thousands of tools and new ones that come out on a weekly basis. And if you're like a practitioner, maybe like a biologist or a clinician or like a application-oriented data scientist, it can often be quite challenging to figure out, which tools to use to analyze your really complex data sets, right? Maybe I only know about like the 3 most common tools, but maybe that might lead me to miss a lot of the insights in my data. So to help us address that, that's why we think, okay, like an AI agent can potentially help the data scientist to try to explore and analyze data in much greater depth, right? And potentially uncover some of the insights in the data that we might have missed ourselves. So that's sort of the goal of this Cell Voyager agent, right? So essentially the agent then will take this input, right, the data set that we're currently struggling to analyze. And also we can tell the agent, here's what we did previously with the data set. And then the output of the agent then will be some sort of new analysis and reports, right, corresponding with figures and also reproducible code to reproduce all of these analysis. And the report then hopefully will contain some interesting new insights, new findings about the data. So briefly, how the Cell Voyager agent works is that first, there's sort of like a preparation stage where the agent will actually look at the data, explores the data, try to understand what is going on in this data set. It can also do a deep research, right, to find how did people have previously analyzed this kind of data before? Like, what are the kinds of interesting questions that people in the community are interested in? And then based on that, then it prepares and initializes sort of the Jupyter notebooks with the relevant tools. So after that preparation, the agents will come up with a set of hypotheses of, here are some of the interesting questions and interesting analysis we could do on top of this data set, right? And based on each hypothesis, it will generate code and then iteratively write the code, analyze the data, interpret the results, and iteratively go deeper and deeper into the data analysis. So here's an example what the data analysis would look like by the agent. So here we enforce the agent to basically do everything in the Jupyter notebook. So everything's actually very transparent and easy for us to reproduce. And the agent basically for each analysis will start with always with some description of what is the actual question it's trying to ask, right? What is the hypothesis it's trying to test? Then it generates the code to implement that test and that hypothesis. Then we'll run that code to generate the figures from the data and then interpret, so it has vision capability. So it would interpret the output figures to understand, like, what did we learn from that analysis, right? And then we'll just iterate through that process, right, until it gets into more and more interesting findings. So the way that we try to use and also to validate this data scientist agent is actually also through close collaborations with human experts. Right, so essentially, we took several papers and very complex and rich data sets that people have recently published, and then we gave those data sets and the paper to the publications, to the Cell Voyager agent, and basically we asked the agent to reanalyze the same data set, but come up with new insights that was not in the original paper that basically were missed by the original authors. And to validate that, we basically will give the output from the agent, this analysis report, back to the original authors and have the authors to help us to validate first that analysis by the agent was done correctly, and then second, that this actually leads to new insights that they themselves had missed in their first initial publication. So we did this through several different case studies. Here I'll just show you one illustrative case study. This is we basically had the agent to reanalyze the previous data set that looked at brain aging. It's from a very nice publication from Anne Brunet's group. So the original study basically looked at mouse brain cells across different ages, and it did single-cell transcriptomics, single-cell RNA sequencing across these different ages. And the original goal of the study is to basically build a machine learning clock to predict biological age from the transcriptome. So that was the original publication. And then the agent basically reanalyzed the same data set and came up with 5 new findings. And then we give the 5 findings to the original authors of this paper. They validated each of these. They found that all the findings were done correctly, all the analysis was done correctly, and then 4 out of the 5 new analyses were particularly interesting and novel from their perspective. So here's an example of the kind of new insights from the agent. Right, so these are basically figures that are created by the agent through its data analysis. So first, the agent would do some standard visualizations of the single cell data, right, and some clusterings. And then relatively quickly, the agent actually asked a pretty interesting question, which is, is there actually a correlation between transcriptional noise and age, right? And the agent actually found that across the different cell types, right, there's actually a very strong and consistent correlation whereby cells that are biologically older tends to have higher transcriptional noise or higher entropy in their transcriptome. Now, this is kind of interesting because this has been reported in other tissues, other organs, but not in this part of the brain before. Right? And the original authors of this paper, when we asked them, they said, oh, they didn't really think about looking at this transcriptional entropy analysis, right? So this was not in their analysis in the original paper. But the agent was able to sort of take this idea that people had shown in other organs, then extend it to the brain and find like an interesting finding. No, it's not too terribly surprising, but I think it is kind of a nice contribution as to the broader literature about the biology of aging. The other thing that's kind of nice here is that the agent is able to actually find other data sets, right, from other publications to reproduce this finding. Here they actually found like another single cell data set from a different publication, and then also analyze that to basically provide external corroboration for this finding to show that there's a consistent trend between transcriptional noise and age, which is also something that we like to see from a nice, robust data science. And then this sort of leads into maybe the third part I want to show, which is this quite recent work that we did that we call paper to agent, right? So the idea here then is to really sort of reimagine what scientific communication and what knowledge dissemination would look like. So the standard way that we communicate science is really through research papers, publications. But if we think about it, each paper is really a passive artifact of research, right? Somebody writes a paper, it's a nice big PDF, and it's actually a lot of work for the readers, right, to read that paper, understand what's going on. And if the paper releases code or data, it's also often quite a lot of work for the readers to figure out how to use the data or how to use the code, right? So that's often sort of a big barrier in knowledge dissemination. So we thought that, okay, rather than having these sort of passive artifacts of research in the form of these paper PDFs, why don't we convert each paper into an interactive agent? So the agent now is the artifact of research, or the agent then will be like a super user of that paper, like an expert of the paper. And the agent can basically help any reader of the paper to understand what is going on in the paper. And if the paper releases some code, the agent is also responsible for helping any user, any reader to run that code on their own problem, on their own data. So then instead of trying to read the paper and make sense of it ourselves, right? So we can just ask the paper agent, okay, can you apply the tools from this paper to my own problem, right? And here the agent then would actually, the paper agent then would be able to then apply the methods, right, from the paper automatically to a new data set, so a new problem, and it will generate all these reproducible codes, analysis pipeline, and provides like a final report in the end, right? So we think that this kind of paper agents then would greatly reduce the barrier to knowledge dissemination, also makes it much easier, especially for a broader range of users and readers, like especially non-technical readers, to be able to really benefit from the insights and the code and the data from these research papers. So briefly, the way that this paper to agent works is that we created this automated workflow to basically convert papers, right, into a robust agent. So in a nutshell, the way we do this is that we build a bunch of different agents who basically goes into the paper and it goes into the code base and the data sets from the paper in depth, and just try to create like a self-contained environment to extract everything from the paper and from its code base. And also, if the paper releases some code with different tools, then the worker agents also extract and build those tools right into and implement those tools. And then they will iteratively refine that environment to make sure it's robust. So this basically creates what people call MCP, or Model Context Protocol. Right? So it turns the paper into this MCP, and MCP have recently emerged in a sort of the industry standard for how to communicate with large language models, right? With external tools. So here, by turning the paper and its corresponding code base into this robust MCP server, this then enables any downstream chatbot, like your favorite ChatGPT or Anthropic models, once it's connected to this paper MCP, then actually really gains all of the deep insights and knowledge about how to use the tools and the insights and data from that paper. So that's sort of the paper-to-agent automated workflow. So just a couple illustrative case studies. So ScanPy is actually one very common tool that maybe many people here in the audience also use for analyzing single cell data. So with paper-to-agent, we can essentially automatically convert ScanPy into a ScanPy agent. Right, so the ScanPy agent then would have access and knows about how to use all different functions and tools from ScanPy. And then you can just, a user can just communicate with the ScanPy agent through natural language. They don't have to download anything, right? They can just ask ScanPy agent, okay, so can you actually, here's my data set, I'll upload this data set. Can you perform some analysis on this data set? And then the agent will be able to actually use ScanPy, right, and perform and generate the downstream analysis. So in this case, right, we basically have agent do their own analysis on this data set, and also have human researchers in parallel to independently analyze the same data set. And you can see that the agents actually produces results that are exactly matches the ones of the human experts. Right? But the human expert here actually requires a lot of experience and this sort of a experienced user of ScanPy. Right? But the agent then would be available to basically a basic biologist, right, who may not have that technical expertise. We also think that this kind of paper-to-agent can be very useful in helping to disseminate new knowledge. So an example of this is that recently, DeepMind released this very nice foundation model called AlphaGenome. So basically, it's a model that can help you interpret the functional effects of different genomic variants, right? But it's not that easy to use. There's publication, also a lot of different software released a part of it, but it's not that easy for the biologist to figure out how to actually use this new framework. Right, so with paper-to-agent, we can also easily create an automated AlphaGenome agent. Right, so now the user, like a biologist, can just ask the AlphaGenome agent, right, so can you actually interpret this variant for me and try to identify causal genes for LDL cholesterol? The agent is sort of like the expert at using AlphaGenome, right, so it looks at that problem and then it figures out how to use AlphaGenome, right?
Yeah. We lost you, James.
Yeah.
He's back.
Oh, okay. I apologize. Sorry, I think the internet got cut off. Can you see the slides again?
Yeah.
Yes.
Okay, great. I was just saying that, so here the AlphaGenome agent basically will do the analysis and use AlphaGenome, right, for the user, right? And then in the end, it will provide like an interpretation and results for the user. And all of the intermediate steps are also saved, right? So the user can just go back and if they want to reproduce any of the findings or modify it, it's easy for them to do that. But I think this is particularly useful because now the user, like you don't have to download and figure out the compute and all that stuff yourself, which can be quite daunting for a lot of the practitioners, right? They can just talk to our AlphaGenome agent, similar to how you would use ChatGPT, and then the agent will actually do a lot of this analysis for you and be able to help you to essentially explain what is going on in the paper. So we're quite excited about reimagined what communication would look like, right? So we basically want to convert all these passive paper artifacts into interactive agents. So that's, imagine in the future, every paper that's actually published will actually have like an agent that's sort of like the expert corresponding author that can help the readers understand and to use the insights and the tools and code from that paper, right? I think that would really greatly make research much more reproducible and also reduces the technical barrier for knowledge dissemination. So I'll wrap up by saying that I think there are also, when we're putting a lot of these different pieces together, and there's sort of an experimental conference we're running, currently running, which is called Science. So this is the first conference where all the authors and also the reviewers are. The motivation at journals that actually do not allow AI as co-authors, right? Which sort of minimize, people to either not use AI or to like minimize how they use AI, right? But we thought we'd flip this around so that for our experimental conference, you have to be an AI to submit to the conference. Humans can be co-authors, but AI needs to be the first author, the primary author to do most of the work. And then each submission also be reviewed by other AI, right? And then the final assessment will be done by human experts, including some really top experts like Nobel laureates in the field. And we're making all the submissions, also all the reviews publicly available. So there'll be a good resource for the community to learn about what are the strengths and weaknesses of AI agents in doing science, like what kind of mistakes do they make? And on the flip side, we can also then use this to learn about how good is AI at actually reviewing papers, like can AI reviewers, what are the biases they have? Can they actually find mistakes in these works, right? So I think this will be a good learning experience for the community. So it's currently running. We've received over 300 submissions, and the conference itself will actually be a virtual conference online next month in October. So we'll be streaming all of that. It'll be freely available. So love for people here to participate. So finally, I think it's also important to summarize some of the limitations of the AI agents. And I think that also really highlights the complementarity with human expertise. I think the current AI agents, they're better at using tools that people have developed, existing tools, rather than creating new tools entirely from scratch. Right? For example, the AI agents, they could use AlphaFold, but they are not very good at creating the next version of AlphaFold. I think that's really where it also complements the human expertise, right? So human experts are very good at going deep into a particular field and being very creative and creating a new tool, right? And then the AI agents can potentially help to apply those new tools to a broader range of problems, right? To help with this research dissemination. And similarly, the current agents I think are better at more breadth rather than depth, whereas the human experts often, can go much deeper into a particular domain. We're also limited by the amount of interdisciplinary breadth that we have, right? So this is also where I think the AI and the human can be very complementary. It's important to note that the agents, they do make mistakes, and often a lot of mistakes happen because the AI doesn't quite fully understand the broader context of the project, right? For example, if you give the AI agent some data set to analyze, but if I don't tell the agents how the missing values have been imputed in the data, then the agent's going to make assumptions that are not valid. That's why I think increasingly it's going to be more and more important for us to try to really capture all the metadata, right, in how we process the data in sort of like more like an AI-friendly way. It's easier for the agents to ingest and to process that broader context. And then that's also why it's really critical for the human experts to continue to review the key outputs from the agents, right, at the key milestones. And finally, but importantly, the agents are currently limited to more in silico analysis. They don't do their own experiments. This is also where I think even closer collaborations with experimental colleagues to do the wet lab validations is even more critical now. Right, so I'll just wrap up here. So I hope to show these 3 examples to illustrate why we're quite excited about building these AI agents as sort of our teammates and partner for biomedical discovery. In the virtual lab, we see how teams of different AI scientists can work together, have their own meetings, and try to tackle quite challenging problems like designing COVID binders that we can experimentally validate. In Cell Voyager, we see how data scientist agents can help us to analyze complex data sets and reanalyze data sets and find new insights that we have missed in our own initial analysis. And with paper agents, we're thinking of this sort of a way to reimagine what the next generation of papers should look like. And ideally, that should, by turning passive papers into active agents, they can really greatly facilitate research communication. I just want to acknowledge, again, my students that led each of these projects. So Carl Swanson for the virtual lab, Sam Albert for Cell Voyager, and Jiachen Miao for paper to agent. So happy to take any further questions.
Yeah, we have a question up there in front.
Yeah, thank you for the great talk. So you introduce many different types of agents, and a lot of them are quite for specific purposes. So why would you say that, for instance, it makes more sense to have one agent for a specific paper instead of one agent that can answer questions on a group of papers?
Yeah, so it's a good question. So what we do is to basically turn each paper into an MCP server, right? So then you can actually, in principle, put all of these different MCP servers from different papers into the same downstream agent, so that you could actually have a single agent that actually knows about all these papers. I do think that it actually makes sense to have like one MCP for at least one paper or for like a series of papers, because each paper, let's say if it releases like often it produces like maybe a self-contained code base, then you want to basically have an agent that's sort of or MCP that's really expert at using that code base.
We're gonna go one online.
All right, so we have 13 questions pre-entered online and a one raised hand. So I don't know how to pick. I'm gonna allow the person who raised their hand to talk, but they lowered their hand. All right, so then the first question actually is from 1228. So it's a few minutes ago. What is the minimum level of human expertise needed to work with these agents?
Yeah, so it's a good question. So I would say you can, for each of these agents, you can communicate to them through human language, right? So the human expert does not necessarily need to do a lot of coding or know to do a coding themselves. But it does still require expertise and some sort of savviness in being able to like, interpret the output and be able to provide useful feedback to the agents, right? For example, they should still need to like look at like the Jupyter notebook produces by a Cell Voyager agent and see like does it actually make sense or not? Does it look reasonable?
All right, we'll do one in here because I got.
Any other questions? We got one way over there, go ahead.
Yeah, so my question is regarding the consensus and the conflict resolution, like how are the personalities built? Is it only skill based or is it also has the human personalities like openness or agreeableness? And how does that impact the process?
Yeah, so it's a good question. Regarding the personalities, like if you look at the default AI scientists, right, they're often very polite and maybe sometimes overly polite. So that even when they get into disagreements and debates, they're somehow like maybe a bit too nice when they have disagreements, perhaps the opposite of humans. But that can also be a problem. Sometimes we also wanted the AI scientists to be a little bit sharper, a little bit critical, so they don't worry about hurting the feelings of the other AI agents, right? So it's not in these publications, but in other works we have been doing so, where we actually try to essentially assign different personalities to different AI agents, right? So sometimes you can make AI agents like more critical or more neurotic, more open-minded, right? So there's these sort of big 5 personalities that we can assign that to different agents. And then we've been doing different experiments to see what combination of personalities actually can lead to more reliable, more creative results, right? For example, sometimes it is useful to have like critics that are a bit more neurotic, and then that can also sharpen the discussions.
Because I get kicked off, the question queue is now gone, so I can't see all the questions. That's a Zoom problem. Okay, someone just added a new one, so unfortunately this is question 14, but because I got kicked off, I had to jump in line. Is paper to agent the future of translational medicine?
I wouldn't go quite that far. I mean, I view I would hope that paper to agent would be a tool to make it easier to translate any research, right? So you can imagine that any research that come out would have an agent attached to it, and the agent then can help to translate that research, right, into into new clinical settings, right? Or help the users to be able to apply that work, right? Because I think that kind of translation from innovation into users is often the biggest barrier for a lot of the work that we do, right? But now we can sort of have the agent to help us do that translation. I think that would really greatly facilitate.
All right, I'm gonna take the last question. So this week, there was an article in the New York Times from Jay Barachata on NIH and the problem that they are just an agency that produces publications, right? And so of all the grants we receive, the number one criteria is how many publications you submit. So how do you think your patient agent tool might shift the paradigm in that situation? Or am I like on the wrong sort of path here?
Yeah, I think that's a really interesting question. I think in many ways, the grant agencies and also community might be a bit over-indexed on the publications, right? Because if you have just a bunch of these like PDFs around, right, that doesn't really help anybody unless there's somebody on the other end that figure out how to use that PDF to do something useful. Right, because the PDFs, they don't talk to each other, like there's no ideas being exchanged, right? And that's sort of the motivation actually for the paper to agent. And instead of having all these PDFs that are passive, now you actually have a bunch, imagine each new research has an active agent, and now the different agents can actually talk to each other, right? They don't have to wait for us to send emails to the corresponding authors. They can just talk to each other. So if somebody actually, let's say, develops a new method, and then there's a new agent for that new methods paper, and some other group has like a new data set, and then there's like a new agent for that data set, right? And then those method paper agent can then just talk to the data set agent directly and then collaborate and then apply the method to the new data sets, right? I think that will actually just make this whole research ecosystem much more interactive and much more fluid.
So we have a minute, so I'm gonna sneak in one more from Steve Goodman. If AI can create something as complex as nanobodies or new hypotheses, why can't they create new tools, particularly, or even improve upon current models? And is it just that we haven't tried hard enough, or ask them to create them?
So that's a great question. I do think that there are some promising signs that the AI now can also start to build new tools. For example, the nice works from it's called Alpha Evolve from DeepMind show that the agents can, by doing a large amount of multicolor tree search, able to actually come up with some of the fastest new algorithms for solving certain kinds of mathematical tasks, right? So that would be an example of the agent really creating a new tool that surpasses any existing human tools. And I do think that's a promising area where the agents can start to actually train their own models and then create new tools. The current generation of agents, even with GPT-5, I think are still much better at using existing tools and maybe making tweaks to them. But perhaps the next version will be better at developing new tools.
All right, well, on that hopeful note, we're at time, and on behalf of Jonathan and all of us in the room, I would like to thank you, James, and hope to continue the conversation with the paper authors in the room.
Thank you so much.
Great. Thank you so much.